Dear friends, welcome to NEET and CET coaching program. Today we are working out MCQs on the chapter Genetics and Evolution. This include 3 chapters actually: Principle of Inheritance, Molecular Basis of Inheritance and Evolution. Let us come to the first question. The number of true breeding plant varieties selected by Mendel for his hybridization experiments in garden P is options 1, 2, 7 and 14. Remember, Gregor John Mendel is called as father of genetics. He was the person who conducted plant hybridization experiments using Pisum sativum. He worked on 7 pairs of contrasting features, that is 7 characters, each with a pair of contrasting features. We call contrasting features as traits. That means he used 14 plant varieties. So answer must be option 4, 14, because 7 characters, each with a pair of contrasting feature. That means if you take height as one character, 2 varieties, one is pure tall variety and other one is pure dwarf variety. In this way, for 7 characters, each 2 varieties, 2 into 7, that is 14 varieties of pure breeding plants. Now we move on to the next question. Which of the following group of scientists first to identify the factor responsible for transformation of R2 form into S3 form in Diplococcus pneumoniae is DNA? The options are option 1, Hershey and Chase; option 2, Avery, MacLeod and McCarthy; option 3, James Watson and Francis Crick; option 4, Meselson and Stahl. Dear friends, again let us look into the question, then we can go for discussion to arrive at the answer. Here the question is about to identify the scientist who discovered the factor responsible for transformation of R2 form into S3 form in Diplococcus pneumoniae. I hope all of you know it is Frederick Griffith who discovered transformation of R2 to S3 form, and we call because of this the transformation as Griffith effect. The only problem with Griffith experiment was the transformation happened in the body of mice. Thereby he was not knowing what chemical responsible for converting the R2 form into S3 form. Later, the same experiment was repeated by Avery, McCarthy and MacLeod. They conducted this experiment under in vitro conditions using digestive enzymes and showed that it is the DNA which is the transformation chemical. That's how the scientists started talking about DNA as the genetic material. So answer is option 2, Avery, MacLeod and McCarthy. When we look into the option 1, Hershey and Chase, I hope all of you know, they conducted the experiment using bacteriophage, where they labeled the protein coat of the virus and also the DNA. For virus, they labeled the sulfur, they used S35. For labeling the DNA, they use phosphorus, labeled phosphorus, that is P32, and they were able to show it is the DNA or the genetic material of the virus is put into the bacterial cell. So we call this as transduction. the credit of discovery of DNA as the genetic material in bacteriophage goes to Hershey and Chase. James Watson and Francis Crick, of course, all of us clearly know about their contribution. James Watson and Francis Crick, I want you to remember here is they are the first to propose DNA double helix or duplex model which describe the structure of DNA. In addition, they also proposed self-replication of DNA by semi-conservative method. Michelson and Stahl, the option 4, what we see here, they are the one who showed that replication is indeed semi-conservative type. So, the answer for the question 2 is option 2. Now, we move on to the next question. Which of the following theory explains continuity of life and not origin of life? Remember, this question is related to the chapter evolution. Here 4 options speak about the theories. Let us look into the option, then we are going to discuss the question and arrive at the right answer. Option 1 says spontaneous generation theory. Option 2, theory of biogenesis. Option 3, theory of chemical evolution. Option 4, theory of cosmosova. I hope all of you are familiar with these names. Spontaneous generation theory is the belief of people which was there till 17th century. People believe that life is going to originate constantly from non-living and lifeless matter. It speaks about origin of life from non-living things and lifeless matter. So, thereby this is not our answer. When you come to the theory of biogenesis, remember theory of biogenesis was proposed by Francisco Redi. Of course, in our NCERT text for 12th standard, the name of Francisco Redi not mentioned. At the same time, you have studied that Louis Pasteur conducted an experiment that disproved the concept of spontaneous generation of life. And Louis Pasteur experiment proved that life come from pre-existing life. What was the statement proposed in the theory of biogenesis by Francisco Redi? Remember, theory of biogenesis is not going to explain about origin of first living organism on earth, what we call protobiogenesis. Origin of life relates to protobiogenesis. So, option 2 is the answer. Now, let us look into the option 3, theory of chemical evolution. This is nothing but theory of chemical evolution of life proposed independently by A.I. Oparin, that is Alexander I. Oparin and J.B.S. Haldane. Also remember the place to which Oparin and Haldane belongs to. Oparin is a Russian scientist, whereas J.B.S. Haldane is a British scientist. An interesting fact is J.B.S. Haldane became an Indian citizen. The most accepted theory on origin of life is theory of chemical evolution. It is also called materialistic theory. Now we move on to option 4, theory of cosmosova. Even today there are scientists who believe in it. The theory of cosmosova is also called panspermia theory. What is this theory about? This theory believes that life was brought to the earth in the form of resistant spores from other planets where life already exists. Now we move on to the next question. The significance of Mendel's dihybrid crosses is, four options. Let me read this for you. The appearance of new traits in F2 plants due to independent assortment of genes. Option 2, the appearance of new combinations in F2 plants due to independent assortment of genes. Option 3, the appearance of new traits in F2 plants due to genetic recombination. Option 4, the appearance of new combinations in F2 plants due to genetic recombination. Now we have to look into this. They appear almost close, but carefully you have to go through the statement, otherwise definitely you get confused. Let me first look into first and second options. The first and second option, only the word that is going to be different is, in first option the word new traits is mentioned, in the second option new combinations. Same again is repeated in the option 3 and 4, but in option 3 and 4 you are going to look into genetic recombination, which was not used in option 1 and 2. In option 1 and 2, independent assortment is used. My dear friends, you know the three laws discovered by Gregor John Mendel on the basis of his experiment on Pisum sativum are law of dominance, law of segregation or purity of gametes, law of independent assortment. When we talk about the monohybrid crosses, we say on the basis of the results of monohybrid crosses, he was able to propose the dominance and segregation of factors. On the basis of dihybrid crosses, he was able to show that each gene control one character, what is called principle of unit character. He also proposed law of independent assortment. Thereby, answer must be either 1 or 2. Let us now look into 1 and 2. What is the difference? Already we know 1 talks about new traits, 2 talk about new combinations. So, let us look into the experiment, what we know about Mendel's dihybrid crosses. He selects 2 characters for his dihybrid cross experiment, like seed color and cotyledon. For example, yellow round seeded plants were crossed with green wrinkled seeded plants. In the F1 generation, we see yellow color is dominant over green. The round seed shape is dominant over wrinkle. In the F2 generation, in addition to parental combination, yellow round, green wrinkle, we also got yellow dissociating from the round seed shape associating with wrinkle, green dissociating from wrinkle associating with round. That means we have not got the new traits, we got new combination. Thereby, the option 2 is the answer. Now remember, independent assortment is nothing but Mendelian recombination. What is this? Genetic recombination. Getting new combination of gene is genetic combination. It is a result of, remember, crossing over, where exchange of non-sister chromatids of homologous chromosome occur, that you have studied in first year PUC or 11th standard in the topic cell biology related to cell division. Now we move on to the next question. During the DNA replication, the complementary nucleotides picked as substrate are, question is straightforward. and it is knowledge based. So what is the question? During the DNA replication, the complementary nucleotides picked as substrates are options: ribonucleoside monophosphate, deoxyribonucleoside diphosphate, deoxyribonucleoside monophosphate, deoxyribonucleoside triphosphate. First option can't be answered because DNA replication refers to self replication or duplication, that is synthesis of a copy of DNA. The first option, ribonucleoside monophosphate, relates to the ribonucleotide, that is a nucleotide of RNA, not DNA. Now we have to look into option 2, 3 and 4. Deoxyribonucleoside diphosphate, deoxyribonucleoside monophosphate. So remember, when we look into the DNA structure, it is nothing but polymer of deoxyribonucleotides. That means each nucleotide is going to have one phosphate. So we think that the answer may be 3, but not so. Remember, replication is an energy expensive process. Here ribonucleosides, that is deoxyribonucleosides, are not picked in monophosphate form, not even picked in diphosphate form, they are picked in triphosphate forms, where 2 of the phosphates are broken to release the energy which is used for establishing phosphodiester bond between the adjacent nucleotide during polymerization. So option 4 is the answer. Now we move on to the next question. This is a simple question, knowledge based question. The life was originated on the earth about dash billion years ago. 4 options: 20 billion years ago, 4.5 billion years ago, 4 billion years ago, 2 billion years ago. So here they are asking you, the life was originated on earth when? In terms of billion years. Then the simple thing, option 1 says 20 billion years. I hope all of you have read in the book that the entire universe was predicted to be originated about 20 billion years ago. So option 1 is for origin of universe. And 4.5 billion years ago the earth was formed. 4 billion years ago the first life was formed. So option 3 is the answer. Option 4 says 2 billion years. First cellular forms have appeared around 2 billion years ago. We know all the 4 options and the option 3 is the answer for this question. Now we move on to the next question. The number of alleles involved in controlling red, pink and white flower traits in Antirrhinum plant species are. I hope all of you remember what is this Antirrhinum plant species. These plants are called as snapdragon plants. We call them also as dog flower plants. Here we have 3 traits related to flower color: red, pink and white. I hope all of you know this is nothing but incomplete dominance or partial inheritance or intermediate inheritance. What is this incomplete dominance is about? Simple, if I have to make a statement, when we are going to cross two pure breeding parental stock differing in a pair of contrasting features. In the F1, neither of the parental traits are going to be completely expressed. Instead, you are going to get intermediate trait expression. Whereas in the F2, in addition to parental trait, you also get the plants with intermediate trait. And this is what we are having in this question. So, we know the allele for red, the allele for white, when they come together, neither of them are going to express their trait fully. So, we called it as incomplete dominance. So, only two alleles are there, one for red flower trait and other for white flower trait. When these two alleles come together, you get a pink flower. Remember, in the F2 generation, both the genotypic and phenotypic ratio happen to be 1:2:1. One red variety, two pink variety and one white variety. If I have to tell you the genotype, one capital R, homozygous, red. Two capital R and smaller, that is heterozygous, not red, not white, pink. And one homozygous, white, smaller. So, answer here is only two alleles, thereby option 2 is the answer. Now, we move on to the next question. In a bDNA molecule of 1,36,000 Armstrong unit length, there is 20% cytosine. The number of adenine molecule in this DNA is, my dear friends, you have studied DNA structure. You also know Chargaff's rule of base equivalence. We know that the two strands of DNA molecule are complementary, where cytosine pairs with guanine by 3 weak hydrogen bonds, adenine pairs with thymine by 2 weak hydrogen bonds. Since wherever C is there, it is pairing with G, number of C is equal to number of G, that is number of cytosine is equal to number of guanine. If cytosine is 20%, guanine is also 20% in that DNA molecule. Together they make up 40%. That means adenine and thymine together make up 60%. Since A pair with T, that is adenine pair with thymine by 2 weak hydrogen bonds, number of adenine is equal to number of thymine, they together make up 60%, means adenine is 30%. 30% of nucleotide of bDNA molecule consuming a length of 1,36,000 Armstrong unit. So, now we should know what is the total number of nucleotides present in this length of bDNA molecule. What is this bDNA? James Watson and Francis Crick proposed the double helix structure for DNA molecule, and this double helix structure what they have described is bDNA. It is the most common form in which the DNA exists. In bDNA, one completed turn, what we call gyre, consumes 34 Armstrong unit length, and each gyre has 10 base pairs. Thereby, the distance between two adjacent nucleotide is 3.4 Armstrong units. So, how many base pairs are present? How many nucleotides are present for 1,36,000 Armstrong unit length? Simply you take 1,36,000, divide it by 3.4 Armstrong unit, and you are going to get the number of nucleotides. And we are going to get 40,000 base pairs. 40,000 base pairs means each in pair, that is 2. Totally it comes to 80,000 nucleotides. Out of 80,000, adenine is going to be 30%. So 30% of 80,000 is 24,000. Thereby option 3 is the answer. You can always work out whenever we are going to look into such questions, always keep a notepad with you and you can scribble down. You find the answer by yourself. That is going to give you a lot of satisfaction and also the confidence what is badly required.